First of all, a couple of introductory remarks. As you will notice, I have taken the liberty of slightly changing the title of my talk. I'm also giving myself mental notes from my media trainer not to grasp this thing the whole talk. Secondly, I think the nice thing about this is that this is relevant to most of the talks that we've heard this morning. This is actually the result of a mega collaboration, stretching back to the early 80s when there was a huge consortium from multiple academic centers trying to find the gene for cystic fibrosis. There's been a huge effort from the CF Foundation to gather data, and we have an unbelievably rich encounter-based database that has been working effectively for the last 20 years, so we know a tremendous amount about this disease. And what I want to talk about today is I'll just give you a brief overview of cystic fibrosis, and then I will show you how we looked at a specific subset of patients with a drug that was targeted specifically to the protein affected by a subset of the mutations. And then give you some thoughts on where we plan to go next with our next lot of therapies. So cystic fibrosis, obviously, orphan disease, about 70,000 people in the world are affected by this, with 30,000 in the US. It is a lethal mutation, and in the 50s, the median survival was about 6 months. And now we've improved it with largely symptomatic care to about 38 years. That means, though, that most people who die of cystic fibrosis die in their 20s. And the lung disease is the main cause of death. So it's a single gene mutation, most commonly seen in Caucasian populations. And there are about 1,800 different mutations that have been observed in this gene. 1,500 of those cause disease. So this is what the protein looks like, which is encoded by this gene, and it's a transmembrane chloride channel. And when you have a defect in that, basically the net effect is that you get mucus plugging at the epithelial surfaces. So it's not only in the lungs, although the lungs are the most commonly affected and they're most primarily the cause of death for 90% of our patients, it also affects the sinuses. It affects the gastrointestinal tract, and particularly the pancreas. Both endocrine and exocrine function are reduced. And for our male patients, it's almost universally associated with the absence of the vas deferens and thus infertility. So what is it that's causing this, and how does it all happen? And this is a cartoon, basically, of what happens on the top at a normal epithelial surface. So typically, epithelial surfaces are lined with a cilia type cell, which beats in a coordinated way to move secretions across, and typically in the lungs, up through the trachea, and then it's expectorated. And you need that defense because you're constantly, every breath you take in, you're breathing in bugs or other toxins, and so you need a mechanism to capture that and clear it out. What happens in cystic fibrosis is because of the dysfunction of that chloride channel, you basically don't have enough of the airway surface liquid so that those cilia can beat in a nice environment. And so because of the collapse of that airway surface liquid, the cilia are effectively all scrunched up and they just can't do what they're supposed to do. So mucus accumulates, there's dehydration, which makes the mucus very thick. And then there's a great opportunity for bugs to grow and live in a nice, warm, relatively wet environment. And that's what causes the lung disease. So let's now turn our attention to the different types of mutation. And I said earlier there are 1,500 disease-causing mutations, and actually new ones are being discovered, although probably the speed with which new ones are being discovered is relatively exhausted, I would imagine now. But we can actually divide these mutations into certain classes. And previously we divided them on the basis of how the mutation affects protein function. And broadly, you can do that into it doesn't make enough protein, or the protein doesn't get to the right place because it's misfolded, or alternatively, there's lots of protein, but when it gets to the surface, it doesn't work properly. So you see those 4 broad examples there. So, and you'll hear a talk from Dr. Hirawat, whose company focuses on the patients who don't make enough protein because they have a stop codon, and so there's no read-through of the genetic code. And how to skip that or fool the gene into actually reading through is the purpose of the PTC approach. The talk I'm going to give now is mostly focused on there's enough protein at the surface, how can we make it work better? And I will touch a little bit on some of the misfolding, which is the second, the sum CFTR column that you see there. And I'll focus on that a little bit to introduce the idea that we can actually improve or chaperone folding through the endoplasmic reticulum more effectively to produce a greater amount of protein at the cell surface. So the clock's not on, and I have no idea of how the time's going, I'm afraid. So I'm looking for a signal from somebody. So how do we segment out this population? And so again, the luxury and this huge mega collaboration is the vast majority of patients in cystic fibrosis know exactly what mutation they have. And so when you go out there and you say, we want to target a specific population within the disease, we actually know who they are, where they are, and also how sick they are, so that we can plan for these studies. Because if you do the math there, the vast majority of patients have this F508 deleted amino acid at the protein level. And so 86% of our patients will have at least one copy of that, and about 45% will be homozygous. This is a misfolding mutation, so it's not the target of our particular drug at this stage. The G551d is probably the most common single mutation example of a problem where there's lots of protein, but it just doesn't work well. And I'll show you in the next couple of slides how it doesn't work well and what our approach was to improve that working. But you can imagine if 4% of our 30,000 patients in the US and similar amounts of those patients in the rest of the world, we're looking at about 2,000 patients worldwide. So how do you track them down? How do you know that they're behaving in the same way that the other mutations are behaving, so that your clinical outcome variables are likely to have the same prevalence and same severity, so that you can actually design your clinical trials? That's the beauty of long-term, encounter-based follow-up registries to which we had access. So there's also variability in the G551d prevalence. So it's known as the Irish mutation. And so whereas generally in the US population is about a 4% prevalence, in Boston, which is a predominantly Irish community, there are a lot of patients. Around Cork in Southern Ireland, the prevalence is almost 20% in the Cork CF clinic. So that tells you, where you go to be successful in recruiting your clinical trials. So back to what the problem is with this particular mutation. Basically, for the chloride channel to function effectively, it has to be open to allow chloride to exit from the cell. And that effectively increases the negative ions externally, which you can measure using patch clamp experiments. And that negative drive of chloride ions means that sodium, and remember, every time a sodium moves across a cell surface, it takes 5 molecules of water. That's the airway surface liquid. So it means that the sodium stays outside rather than coming back inside the cell. So you want your channel to be open. And this shows in the 2 panels on the right as you look at it, that in the normal CFTR, the channel shown by those red dashes and the high bars of the activity is it spends a lot of time being open. The G551d mutation spends no time being open. So what can you do about that? Well, you can add this ivacaftor molecule, which is known as a potentiator. In other words, it improves the function of existing CFTR protein. And you can see from this experiment shown in the potential difference trace on the left-hand side, that entire trace is moved up to the red dotted line in the bottom panel. And if you look at the probability of the channel being open on the right-hand side, you can see in the clear histogram very low amount prior to drug treatment. And after application of the drug to this system, there's clearly an increase in the negative ions external to the cell, indicating that the drug has activity at the cellular level. So then we know that our drug works, at least in the lab, but these are experiments which are done by taking the cDNA and transfecting it into a rat thyroid cell, and it gives us the proof of concept, but it doesn't actually translate to will it work in humans. And will it work in humans means that we have to move away from looking at the actual function of the channel from a pharmacodynamic point of view and seeing if we can translate that into actual clinical benefit for patients. And so the common things that you look at in cystic fibrosis as outcomes, which have been used as approvable endpoints, primarily pulmonary function, and we look at the forced expiratory volume in 1 second, which is how much air you can blow out in the first second that you do a forced expiration maneuver. We look at pulmonary exacerbations, which are periods where there's acute worsening of the lung disease requiring intervention with antibiotics. These are problematic because they tend to be very subjective outcomes, but you can apply algorithms that you impose on the physician community during the clinical trial so that you get a relatively consistent interpretation of an exacerbation. Weight gain, again, the pancreatic exocrine function results in undernourishment of most of our cystic fibrosis patients, and many of them have BMIs in the 18-19 range and could well benefit from improving their nutrition. And then we have a disease-specific quality of life instrument, which was actually developed in France and then translated over to the English language. So this is the huge value that we get from this mega collaboration and from the work that the Cystic Fibrosis Foundation has been doing for more than 20 years. They've invested a vast amount of money into this encounter-based registry. And on average, those 26,000 patients who are in the registry have about 4.6 encounters recorded in the registry. So you can imagine that if you're allowed access to that, it's a huge, rich harvest for you looking at. And so we knew going into the study that 91% of all patients had been genotyped, so we knew that we could probably find the 2,000 patients, well, the 1,000 patients in the US that we needed. We knew also, very importantly, that the natural history of this particular mutation was the same as, and I show it here, was the same as the most common type of mutation, which is the F508 deletion. That's very important because if you're doing sample size calculations, particularly for things like how quickly you expect pulmonary function to decline, what the baseline pulmonary function is likely to be, how frequently the exacerbations occur, so you can appropriately sample size your study, you need to know how those patients behave. And knowing that the rates of those important outcomes are likely to be identical to the general population of cystic fibrosis was extremely important. As an aside, it was also extremely important when we went through the regulatory approval process to be able to convince both the FDA and the EMA, and subsequently other countries that we've been working to approval in, that these patients are in fact a specific class of patients, and secondly, that we know what happens to them so that when we see an effect from our drug, we can tell that it's a relevant effect. So this is our program, basically. We had a phase 3 study in patients aged 12 and over, a phase 3 study in 6 to 11-year-olds, which was a very much smaller study. There were only 56 patients in that study. And then we were asked to show that actually our drug didn't work in the most common mutation. And so this gave us the double advantage of being highly specific in the indication, which with an extremely small safety database made the regulatory agencies very comfortable because they knew it couldn't be used and it just would not be used in the larger population. And secondly, it allowed us to increase the safety database by studying the patients with the homozygous F508 mutation. So these are the results of the studies, and I'm not going to go into them in great detail except to point out that the pharmacodynamic endpoint in this population pushed patients to below the diagnostic criteria. So we use sweat chloride as the diagnostic criteria for cystic fibrosis. If it's elevated, and in these patients it was elevated to 100 millimoles per liter, if it's below 60, you're not considered to have cystic fibrosis. And so, most extraordinarily, we were able to push these people below the diagnostic criteria for cystic fibrosis. Very interesting, useful pharmacodynamic endpoint for us, but no clinical benefit from that. The pulmonary function, the weight gain, and the almost 50% reduction in exacerbations, clearly huge benefit. And just to put that pulmonary function gain in context, if you actually measure that in cc's of lung volume, on average it was about 366 cc's. And for the ladies in the audience, that's about as much air as you breathe in with a normal tidal breath. So it's a lot of air, and resulted in some amazing and very heartwarming anecdotes, one of which there was a young lady who was quite severely affected with her cystic fibrosis, basically short of breath with any exercise. And we heard this lovely story about how during, not this winter, but the last winter, there was a major snowstorm. And so she said, well, I'm feeling okay. I'll shovel the snow off the driveway. And her husband looked out, and lo and behold, after half an hour, she was shoveling the snow in the neighbor's yard as well, because she felt so good. That's not a label claim, by the way. And we were lucky in that the drug was relatively safe. There are clearly some adverse events associated with it, most commonly headache, upper respiratory tract infection, nasal congestion, rash, and some dizziness. But by and large, the serious adverse events were relatively few and far between, with the one which is interesting, which was hypoglycemia. And we're now actually looking to see whether or not that's actually a drug-related effect on the endocrine function of the dysfunctional pancreas. So, in summary, we had substantial change in our primary endpoint. The lung function improved relatively early and was maintained over a period of 48 weeks, and we had no clinically important safety concerns. Right, now I have to get through the rest of the talk in a very short time, so excuse me if I go fast. So, with that basis, where did we go from there? And the most immediate and obvious stepping stone was that there's a number of other mutations who have exactly the same dysfunction in their protein, which were obviously the low-hanging fruit for us, if you like. The problem with this group of mutations is altogether those 9 mutations constitute 1% of the population. And this is where some of the the new thinking and the changes in the regulatory review have helped us tremendously. And so we were able to go to the FDA and say to them, look, we've established with this classical, if you like, approach to clinical trials that the group who have the G551d mutation are an exemplar of the gating class. We can't do the same study in this incredibly small population, but having established that the exemplar of the class is likely to respond to our drug, can we use innovative drug design to actually test the short-term efficacy in a crossover design so that we can determine that these other mutations also have the potential to respond to the drug. And actually, by using the crossover design, and some of these things like the G551S there's one example that I know of in the US. So these are extraordinarily rare mutations. But we've actually managed to complete a study where we recruited at least one example of all of those patients. And by having the crossover design, we're actually able to look at each of those mutations individually, as well as the extension of the class as a class. And so that's sort of where we are with our regulatory strategy. And we hope to have that sort of data in the relatively near future. So will they respond? The answer is yes, they will respond. So this is Fred Van Goor and Paul Negulescu's work showing in that Fisher rat model that the drug is responsive in the lab. Okay, so then I told you that we'd done a study in the F508s. We did not expect any of these patients to respond. And there are a number of other mutations which fall into the same class. And what you see here is the pre and post, effectively, of treating those patients, treating those mutations with Kalydeco in the lab, and you see that there's no response. So what's the approach for those patients? And just very briefly, I'll share with you the other studies that we're about to launch, which are primarily aimed at patients who are homozygous for this F508 folding mutation. So the principle of this is a double whammy, if you like. So if you could massage or refold or allow that misfolded protein not to be taken out by the endoplasmic sort of quality control system of the cell and allow it to be expressed at the surface, it's still dysfunctional. But actually the nice thing is that it's exquisitely sensitive to the ivacaftor molecule. So if you got some of it there, you could potentiate it quite nicely. So this is the theory behind the combination therapy. So that's the way we will be managing this larger population of patients. There's another group of patients who have what is called residual function. And again, that would seem like a group of patients where they have some function at the cell surface and therefore increasing the amount of that function with the existing drug of ivacaftor would seem to be an approach. And we're pursuing that now. And again, the laboratory data tells us that that is likely to be successful. So a very quick whiz through a fairly extensive program, and I apologize for rushing it. But Dr. Lecan did say that he'd like to save some time for questions. So in summary, basically, cystic fibrosis is a disease that can be characterized by the effect of the genetic mutation on the quality and quantity of the cystic fibrosis protein. And the potential of effect of molecules of drugs can be determined by defining the in vitro molecular phenotypes of those mutations. We were fortunate that the first drug that we looked at in the targeted population of G551d had such dramatic results, and we hope to be able to extend that to the other mutations. And as I said, we have a plan for dealing with combination therapy for the vast majority of patients who have the F508 delete mutation. So thank you very much indeed for your attention. I'd be happy to take any questions.